Hi everyone, my name is Francesca Susca and I'm Pet Development Manager within Lallemand Animal Nutrition. With this presentation, I will show you the multiple benefits of yeast and yeast derivatives products for pet nutrition, and I will show you the importance of selecting the right yeast for the right application. The global yeast market has seen a massive growth over the last few years, due to an increased consumption of processed food, alcohol intake, but also thanks to the greater awareness of the biological properties of yeast. Just for giving you a few numbers, nowadays is used for the annual production of something like 60 million tons of beer, 30 million tons of wine, and so on. Yeast are natural microorganisms with a great biological activities, and so they have a great potential for several applications. One of these is for sure animal nutrition. But let's see what actually yeast are. Yeast are microscopic fungi with a nuclear membrane and a cell wall. The cell wall is mainly made of glucans, mannans, glycoproteins, while the intracellular components are amino acids, peptides, nucleic acids, and so on. The combination of these components make yeast very valuable nutritional supplement as well as good as nutraceuticals. Animals have been fed yeast since a long time, more than 100 years. Actually, it's quite common, the usage of yeast as a feed ingredient, feed additive, for both poly and monogastric animals in order to maintain and improve their health and well-being. But yeast vary a lot because of their metabolism of substrate, their reproduction, their biological activities and properties. And that's strictly linked to the yeast strain genome. So exactly as for bacteria, also for yeast, it's important to identify the strain genome. But honestly, identifying and classifying yeast is quite a challenge because there are more than 60 different genera of yeast, which comprise more than 600 different species of yeast. Of course, they are not all of industrial importance, but Saccharomyces is for sure one of them. And thanks to the DNA homology, has been identified 4 different species of Saccharomyces. And Saccharomyces cerevisiae, thanks to its very extraordinary fermentative properties, is the predominant microorganism used in several industrial applications. There are thousands of strains used in enology, bakery, brewery, savory ingredients industry, while there are specific strains that has been selected because beneficial in polygastric animals, monogastric animals, and there is also the opportunity to make association of different yeast strains in order to achieve a specific benefit. In animal nutrition, the cell yeast can be used as a whole cell yeast that can be live or inactivated. You see in this table we have summed up a lot of information, and I'm not going to go through all of them, but I want just to show you that the identity and the composition of the cell yeast is strictly linked to its functions. If we look at the whole cell yeast live, Saccharomyces cerevisiae boulardii is without any doubt the most well-known and well-recognized probiotic yeast in both human and animal nutrition. It has been isolated for the first time in 1920 from leach infusion, and it has been recognized as a probiotic in human medicine more than 50 years ago. While in 1993 it has been registered for the first time in Europe as a probiotic in animal nutrition. Today is targeted and marketed in more than 60 different countries. There are hundreds of research on the beneficial effect of boulardii in human medicine, but there are also some quite interesting and significant scientific evidence on the beneficial effect of boulardii in dogs. If we look slightly deeper in the mode of action of Boulardii in pets, we can see that it acts at 3 levels. First of all, on the intestinal microflora by reinforcing the positive microflora, but also at the level of the intestinal epithelium, where it is supporting the protection of the intestinal epithelium by decreasing its permeability and increasing the mucus production. But it also acts at the immune system level. Whenever he is able to support the immune system, thanks to its anti-inflammatory properties and also the ability to recruit immune cells. So Boulardii is effective in the area of digestive care and immune support. Whole cell yeast, as I said, can be used also as inactivated. In this case, they are mainly used as nutrients because it's known that yeast are naturally rich in high quality proteins, vitamin of the B group, amino acids, but there is also the opportunity to enrich yeast with specific minerals and vitamins. A good example is the selenium enriched yeast. The yeast is grown on a specific amount of inorganic selenium, and the yeast, while growing, is able to absorb and biochemically convert it into the organic form, which means selenium methionine, selenium cysteine, or several other selenium compounds. Selenium is quite an important micronutrient for pets because it's involved in antioxidant immune thyroid functions. But the whole cell is inactivated, they can even be used as flavor enhancers because they contribute to the umami flavor. And the main yeast used with this scope are the primary grown, the torula yeast, and the brewer yeast. Another big family of yeast derivatives products are the yeast fractions. So in this case, it's used just one part of the yeast cell. Can be the inner part, so we have the yeast extract, or could be the cell wall. And in this case, we're going to have moss and beta-glucans products, or even association of specific yeast fraction that has been specifically selected for their complementary activities. Again, the same table I showed before for the whole cell yeast, in which you can see how identity and composition of the yeast are strictly linked to their functioning. Yeast extracts are the remaining part of the cell yeast whenever the cell wall has been removed. They are used as flavor enhancer, flavor contributor, because they are rich in glutamic acid. They have a content in glutamic acid which is around 6%. But the same components responsible of the flavor enhancement, they are also of high nutritional value. A good example are nucleotides, the building block of the nucleic acids, which are quite important for pets, for their health and well-being, because daily the body requires to build several new cells. And for doing that, he needs to build new RNA and new DNA. So during the period of recovery, but also during maintenance, it's good to supplement pets with nucleotides. Within the yeast fractions, we have the products coming from the cell wall. The most common on the market and the most widely used are mannanoligosaccharides and beta-glucans. But why such a wide usage of those products? On one side, mannanoligosaccharides, they are well recognized to be able to agglutinate harmful bacteria, avoiding them to colonize the gut. In this way, they have an indirect prebiotic effect because, of course, they are favoring the colonization of the gut by the beneficial bacteria. On the other side, beta-glucans, through the activation of a single receptor, the dectin-1 receptor, are able to stimulate phagocytes, which become more active, more ready to include, digest, destroy pathogens. And during this process, signaling molecules such as cytokines are secreted, stimulating the immune response. Usually, the cell wall products are defined based on the biochemical quantitative composition. I mean, percentage of moss, percentage of beta-glucans, and proteins content. But is really all around that? I would say no. I would say that those are important parameters, but they are not the only one. There are some other characteristics, such as the composition, the structure of the yeast, the repartition of molecule along the cell wall yeast, which make the difference, which give to the specific yeast strain specific properties. And additionally, also the production process, we will see it later, has an effect on the specific properties of the yeast strain. The atomic force microscope is a very powerful tool which enables to take a closer look to the yeast cell surface, to look into the biochemical and biophysical composition of the cell surface. Based on a standard microscope, the atomic force microscope is equipped with a specific special case to read the surface of the yeast, reproduce it in a 3D image on the computer. There is a metal tip that travels through the cell wall surface and lifts all the asperities, giving them a 3D images, as I said, and it looks like the surface of the moon with a lot of bumps and craters, which is the topography of the yeast cell. And as you can imagine, the topography of each yeast strain is different, and that's directly linked to the properties of the strain. Furthermore, there is the possibility to look into the interaction between cells. And our scientists have used this possibility to look at the interaction of specific yeast strains with the molecules that were mimicking the action of pathogens. In this way, we have been able to see how specific yeast strains were interacting with the pathogen, which was their adhesion strength and also their point of rupture. It allows to select specific strain accordingly to the adhesion properties. As I said earlier, the structure and composition of the cell wall are not the only characteristic affecting the properties of a strain, but there is also the production process. As you see in these images, the autolysis process has an impact on the cell surface roughness. And in these two additional images, you can see how the cell surface look like before and after autolysis. So there is an impact of the production process on the yeast properties. Through a 3-steps project, our scientists have been able to select specific strain with different properties but complementary activities. So they have started by developing a new method of characterization of the yeast using the atomic force microscope. And thanks to that, they have selected the best strain depending on the effect they have seen. Finally, they have applied to these strains the optimal industrial production process. That's how YOUNG is born. YOUNG stands for Yeast Association New Generation, and is an innovative product on the market of yeast because it's the very first formulated yeast on the market. It is made of 3 strains that have been specifically selected in the way I just explained to you, and the strains have been selected and combined in order to provide superior binding capacity and a synergistic, broader and much balanced immune modulation. The beneficial effect of such an association of yeast fractions looks similar to the beneficial effect of the cell wall products. But actually, the high prevalence of adhesive polysaccharide patches on the yeast strains that has been selected gives to YOUNG stronger pathogen binding capacity. And on the other hand, the combination and the formulation of Yang, thanks to the selection of the three different strains of yeast, permit a simultaneous activation of several immune receptors, which result in a broader and balanced immune modulation. The superior binding capacity of Yang has been evaluated as first in vitro on human cells mixed with Escherichia coli and Yang. 1 hour after the infection, we have looked into the inhibition of Escherichia coli binding on epithelial cells. And if you look in this graph, you can see that along the increasing dosage of Yang, you can see a decrease in the percentage of coli binded to epithelial cells. That support the superior binding capacity of Yang, but also is showing a great dose effect correlation. The same concept of superior binding capacity of Yang has been validated also by comparing the binding capacity of 0.08% of Yang versus 0.2% of a wood moss product on the market. We looked at the binding capacity versus 5 strains of Escherichia coli isolated from the dog and cat feces at different life stage. All the strains were collected during episodes of enterite, and they were identified as the unique causative agent of diarrhea. What we have seen is that Yang at a much lower inclusion at 60% less was leading to a significantly higher Escherichia coli agglutination. If you look in the graph, you see that the Yang group was able to agglutinate 19% more Escherichia coli. Coming to the immunomodulation, to the broader immunomodulation property of Yang, it's also this property linked to its formulation. Several studies have demonstrated that the activation of the immune response by a single receptor route, like as I showed you before, beta-glucans and dectin-1 receptor, may cause an overstimulation which can lead to an pro-inflammatory status of the dog, which has, of course, a negative impact on the animal. On the other side, Yang, being formulated and containing different strains of yeast, is able to be recognized and so activate multiple receptors on the gut, resulting in a synergistic immunomodulation, which avoid the risk of overstimulation. We have validated this concept by comparing the level of activation of the immune response of the 2 strains of Saccharomyces cerevisiae versus the strain of Ciberlindera yadini versus Yang, so the combination of the 3 strains together. And as you can see in this graph, you can see the effect, the cumulative effect of Ciberlindera yadini, the 2 Saccharomyces cerevisiae strain, and then an additional effect given by the 3 strains together as by the formulation of Yang. These great results is due not just to the fact that the 3 strains of Yang has been rightly selected and combined, but it's also due to the fact that they are able to simultaneously activate multiple receptors on the gut. We have validated this concept also in vivo in different species, and one of them is dog. I'm pleased to show you here that we did an in vivo trial to validate the synergistic effect of Yang on the immune system. And we work with 16 healthy adult dogs in a trial that lasted 28 days. And we, among several other parameters, we looked into the fecal immunoglobulin A, fecal EGA. And as you can see here, the fecal EGA resulted to be significantly higher already 21 days after supplementation, and there was 59% higher level of EGA at 21 days and 56 at 28 days. Just as a reminder, EGA are a well-known and well-recognized non-invasive biomarker of immunomodulation, and there are quite a lot of available publication on the measurement of fecal EGA in different. EGA is the most abundant class of antibody in the mucous membrane, and it is important in mucosal immunity because it inhibits the attachment and penetration of bacteria in the lumen, increases the mucus secretion, and prevents inflammatory damages for the gut epithelium. So I'm back to the table I showed in my earlier slides. In here I put the two family of the yeast derivatives product, the whole cell yeast and the yeast fractions together. Probably now this table, I'm sure, makes much more sense to you. Again, I'm not going to go through all these details, but I want to show you once again that the identity of the yeast and its composition has an effect, has an impact on the functions that the yeast have. And here you can find also some additional details in terms of dosage, average dosage of these yeast derivatives products, and also some information on their stability to heat treatment and few tips on the regulation. So we have seen that not all the yeast are the same and that their biological properties are directly linked to the yeast strain genome. Furthermore, there are several factors, amongst them, of course, structure, composition, and production process, which can affect the yeast properties. But knowing the mode of action and how the yeast are expected to work in the animal, it's quite important because help you in selecting the right yeast for the right application. So I hope I have been able to give you a good overview of the great potential of using yeast and yeast derivatives product for pet nutrition. And I want to remind you that from flavor enhancer to source of nutrients to functional ingredient, yeast are versatile, natural ingredient to bear in mind whenever you are formulating for the beloved pet. I thank you very much for your attention, and if you have any question, please don't hesitate.